Well, thank you very much for the kind invitation and the chance to give a short update about the case that we have deal with. Actually, it was the second one. The first one was grape. And although most of the plants has been sequenced until now are homozygote genome because they have inbred lines like also barley, but not only. So we have this goal to deal with really cultivated variety in fruit plants, fruit trees mostly, especially the ones which have self-incompatibility. Either we are able to produce double haploid in vitro from some type of artificial in vitro culture, or there is no way. So actually, grape was propedeutic of apple. So in this sense, I have prepared a talk divided more into in two sections. One more technical, one more related to the results we have produced and published recently, just to show you where we are located for the ones that do not know where San Michele is. San Michele Adige is the last village in province of Trento, just below the Bozen region, the Bozen province, and is mainly based, our activity is mainly based on grape and apple, also soft fruit, especially strawberry and raspberry. So, well, actually, the last year was the year of the Rosaceae. Well, 3 Rosaceae, like peach, apple, and strawberry, has been sequenced, and the sequence has been released in the public domains. And it is actually, in the botanic sense, the first family which has such a large number of plants, following also the raspberry and the pear, which is also going to be sequenced, will be the first family, botanic family, having 5 genomes which can be then compared to each other. And like the Triticeae, which is very well studied because obviously the importance as a food for the whole planet, the Rosaceae has a very interesting aspect in comparative genomics, which will be just touched at the end of my talk. Why the apple genome? Actually, in Trentino we have 60% of the production of apple in Italy, so it is clearly the most important cultivation in our region. And why Golden Delicious? Actually, because 85% of apple cultivated over there is Golden Delicious. We have the luck that this variety has been produced in US, but has found the ideal environment exactly in Trentino Alto Adige. And over there, it releases its own flavor, the best quality of this apple. Then there are other few advantages. There is also a double haploid produced in France, which is a relatively rearranged genome because it has been passed through a couple of self-pollination, and then the rearranged genome is not exactly the ones that we have it, but it helps to define the haplotypes. The genome is relatively small in comparison, especially with the barley, as we have heard it before. And from the tackle the problem was for us very important to pass from the Sanger process to the 454 data. And actually, this was an experience we have already had in grape. In October 2006, actually, we had this approach, which was mainly based on Sanger sequencing, with some addition of Sanger sequencing with some addition of 454. When 454 was producing roughly 120 bases of each run, and no more than 100 million of bases per single run. And it was actually not enough to support a large genome like a plant genome, and also it was mainly based on as a tool to fill in gaps, which were obviously for an heterozygous genome pretty complicated. But it helped quite a lot. And we passed from the grape experience to the apple experience, where 454 is actually dominating the sequencing, and the Sanger sequences was really just a scaffold, a sort of skeleton. Just for the people that do not know, sorry, to go back here. Yeah. For the people that do not know exactly the processes, by the Sanger you cannot decide, you cannot avoid to miss some part of a genome because it's not clonable. And obviously the big advantage is the size of the reads, but the big disadvantage are the length of the time that you spend to do this kind of runs, for instance. While by 454 the length was relatively even smaller than that. Nowadays it's clearly stable around this size, and nowadays it's easy to reach this amount. At that time was not possible, but to do the same kind of coverage you need 1/10 of the time. And the other advantage is the fact that there is no need to clone the DNA, so you share the DNA, you divide it, so the entire genome is represented in your sequencing. So these were the results from Sanger, and I don't go in details about the approach, the technical approach. But nevertheless, for grape it was, as I said, just a gap filling because the small size of the fragments, and nevertheless it helped to close 25,000 gaps within the first assembly, and it was useful to extend 34,000 of contigs. And that was pretty good as a result, and that convinced us to approach in a vice versa way. So focusing more on 454, the entire apple genome, although it was a bit bigger, it's 1.5 the size of a grape genome. In the 2008, as I said, there was the introduction of the titanium chemistry. The titanium chemistry actually was essential to produce such kind of amount of reads, and also the pair reads of different size of libraries, which were actually resolving most of the problems in terms of assembly. What means pair reading? It's the way that the 454 sequence is actually sharing the DNA. But if before to share the DNA, you prepare libraries of fragments extracted from a gel, of 2 as well 20 kb in length, you know the size of your fragment, and then you can ligate your fragment of known size with a small insert of DNA, which is here indicated, that you know as a sequence, and then you divide, you break your DNA, and when you sequence this part, the big advantage is that you may lose a small amount of sequence clearly, but the big advantage is that you know exactly the distance that you have in your fragment between the forward and reverse, between the two extremities of your fragment. That means when you go to assemble, and you know that this fragment is 20 kb, when you assemble, the fragment, the sequence here and the sequence here cannot be in a different distance than 20. It cannot be 5, it cannot be 100 kb. It must be 20, and then you can force your assembly pointing on these results, or vice versa, you can exclude that your assembly is correct if these assembly do not respect the size of your fragment. And then actually we obtained this amount, and 11.4 X coverage was used for the assembly. Assembly genome is a never-ending puzzle, and people like Giorgio Valle or Neil knows now very well that this is the old process with Sanger sequence, but the principle is not that much different. You fill, first of all, you assemble small contigs with enough overlapping sequences, and then you increase by the size of your fragments using the ends of the sequence of the fragments which overlap but bridge the two contigs to extend the scaffold, and then using back clones, most of the cases, you can join groups of contigs with some gaps in a much bigger scaffold, which then represent a discrete size of your chromosome. Dealing with heterozygous genomes was a real nightmare, and that was for us an experience when we did, together with the Udine University, the physical map of the Pinot Noir grape. I show you here just an example of the physical mapping of the back clones to reconstruct physically the back by back when we were still deciding if to proceed back by back or whole genome shotgun before to begin the grape sequencing. And actually the FPC program, which is perfect to align and to reconstruct the physical map of back clones, reconstructing the chromosome physically of a certain species, is not suitable actually to work with the heterozygous genome when it contains a lot of SNPs and small inserts, indels. So we sequence the seven backs here indicated in blue, almost completed apart the small gaps which then were filled in by primer working. And this figure represents exactly the position I showed before in the contig produced by FPC. When we went then to sequence completely with the 6x coverage by Sanger of these fragments, actually the backs were then piled up in a pretty different way with a gap in between. So this overlapping was not real, was not really existing. And the two groups, the two contigs, were then grouped in a different way in a much shorter fragment. That means the physical FPC mapping of the Pinot Noir was 1.6 the size, the real size of the grape genome. So by developing proprietary programs, we were able to detect sequencing error from true polymorphism among the two haplotypes. Unfortunately, these kind of softwares are proprietary of Myriad Genetics, but nevertheless, they were extremely useful to call among a great number of individuals of our maps, of our crosses, directly the real polymorphisms, helping in the assembly of the two haplotypes, recognizing real from wrong SNPs. But nevertheless, the big advantage, having a large amount of SNPs and small indels, was on the contrary, from one side, it represents a problem to assemble a genome. From the other side, it was extremely useful to link our genome to the maps, pretty large maps, of the 19 chromosome of grape or the 17 chromosome of apple. And it was pretty easy to choose something like 2,000 SNPs, well distributed along the 17 chromosome of apple, choosing among over 2 million of SNPs truly detected by the methods that I show, the softwares I showed above. And then for each single linkage group was a quite large distribution of markers among the SNPs and marker satellites to recognize the chromosomes, so the pseudochromosomes, with a coverage that at the end was roughly 72% of the genome anchored to the virtual chromosomes. So the pseudochromosomes are here, indicated in red. Clearly, most of the, so we have chosen to represent here only part of the markers to avoid an excess of density, but these are selected markers among the 1800 markers screened on 6 progenies. 3 of them contain golden delicious as a parent. In blue I indicated the scaffolds which are also oriented, because clearly if you have one single, in a contig, you have one single markers, you may orient your contig either or in this direction. If you have 2 markers or more, then your position is clearly linked to the others' position along the pseudochromosomes, and then you can be sure that your scaffold is correctly oriented. The biggest, obviously, have orientation. In yellow are not oriented chromosome, and in white are the scaffolds which are most likely belong to only one of the 2 haplotypes, because quite a lot of DNA, roughly 10 to 12%, is disposable DNA. That means it has been represented very well in other presentation by several other colleagues in genomic congresses, that it happens often that plants has a quite plastic genome, and if you compare 2 haplotypes, as in this case, within one variety or embedded lines, often you find a certain percentage of DNA which is actually typical only of this embedded line, and is so-called disposable DNA, because it doesn't seem to be essential for the surviving of a plant, but it characterizes a certain type of variety. And then some pills of the publication we have done last year in October about the apple genome. This is more scientific and less technical. One of the answer we try to give to the community was to definitely close the discussion about the origin of the 17 chromosome of apple and pear, which are in the Rosaceae within this subtribe, Pirae. The only subtribe which contain such a number of chromosomes, which are almost double than the most common numbers in the Rosaceae groups, which are actually 7, 8, or 9. The discussion was, or the proposal for many, many years was that the 17 numbers, the 17 chromosome number of apple and pear was derived by the allopolyploidy between Spirea and Prunus. Prunus is the family of, is the genera of peach or apricots, for instance. And Spirea is more represented in North America botanical garden. So these 2 representatives were the most believed, let's say, ancestors of this number. Actually, from the phylogenetic point of view, Gillenia, which is at the base of the tribe of Epireae, is actually the most likely believed ancestor, because although the number of chromosomes then would be 18, but then we will see why it is there is a loss of 1 chromosome. But nevertheless, the most close phylogenetically actually was this kind of genera. And the second question was where it's coming from the modern domesticated apple, which is until now believed to be a sort of inheritance between the Malus sylvestris, the European wild apple, and with the participation of some of Asiatic Malus species. But actually, Malus domestica group then together with the Sieversiae, which is actually from Northern Asia Malus species, and it has nothing in common, or actually it's close, but is not mixable with the Malus sylvestris, which is then deduced to be almost not contributing to the present Malus domestica cultivated in Europe. And it was a pretty small shock for people working in the fruit field. Another aspect that we have faced during the time that we were initially assembling the genome was the surprise to see that several chromosomes were homologous pair by pair. That means we have seen that the synteny of these bars are genes, and these different colors are different gene position. That, for instance, like here, chromosome 13 and chromosome 16, the position, not only they were the same genes, or at least orthologous, presumed orthologous genes in a synthetic position, and they were extremely conserved, like they would have been the same chromosome. And this was somehow answering to our surprised calculation of the number of genes which have been found in the apple genome. Actually, we have seen in most of the cases plants having between 25 to 35 genes in their genome. Apple has almost 60,000, and we were pretty surprised. We are thinking, well, something has to be wrong. But actually, looking at this, then it is less surprising that such kind of number, it may be double than peach, for instance, which has half number of chromosomes with obviously a single or only one of these two as a chromosome in his own chromosome number. So here are represented some other homologies between chromosome pairs or chromosome arms. And this brought us to the idea to develop this evolution in terms of the apple genome. These ancestors with 9 chromosomes was then the base of the development of most likely an autopolyploidization in the apple genome, having either pair by pair recognizable chromosome or arms by arms, with a loss, and this was a bit more recombined, a loss of one of the chromosomes, the 18 chromosome, which has been fused partially within the chromosome 15, which is actually double in size with respect to the other chromosome, which are almost all the similar size, between 20 to 25 million bases. And chromosome 15 is 48 million bases, almost double than the others. And some other pills actually about peach and apple. As you can see, although we respect the publication of peach genome, so it's a comparison between the map, the most developed map of peach compared with the apple genome, but nevertheless all these genes have been used to develop markers, and actually their synteny between one chromosome of peach and chromosome pairs of apple is extremely well maintained and easy to be identified. And here something happened between IBM and Apple. Actually, there was just one row, but nevertheless, here we give some more details about apple and peach and apple and strawberry, which gave actually similar results with easy identification of the ancestors of common ancestor between all the rosaceae, which were most likely with 9 chromosomes, and ancestors with 9 chromosomes, which have been given rise to the 8 chromosome of peach, 7 chromosome of strawberry, and the 17th present chromosome of apple. So this is actually published on BMC of Molecular Evolution last month. So you may have some more details with respect to this conclusion. So last but not least, the list of the people, actually not all of the people there, because there are many more, but all the people that signed the publication of apple and strawberry, which has been published in Nature Genetics again in February this year. And that's all what I have to say to you. That's complete.
Full in time for questions. Thank you, Riccardo.
Last speaker of the day is always
no questions. But
the killer.
If nobody does a question, so did you mention double number of genes? But do you have any evidence of expression, or I mean, they are all working, or pseudo genes, or?
Well, from let's say the data coming from the sequence, there seems to be real genes. So pseudo genes has been already eliminated. We have worked with several groups also in Europe and in the US, try to decrease the number of predicted genes. The lowest number we were able to produce is 57,000, actually still almost a double of the other genomes. We do not know actually the redundancy between, let's say, the genes present in chromosome 13 and the genes present in chromosome 16. We are now working on trying to understand if they actually are real redundant or they are not. So if it behaves as a false tetraploid or a real diploid, the inheritance is disomic, so it's actually a normal genome, diploid genome, but nevertheless, it is, and it will be very interesting to have a look to the transcriptome and to the chip, microarray chip we are now preparing with nimble genes, so it would be nice to see what is what.
Okay, thank you. Thank you, Riccardo.